Hey guys, my name is Isabella Pedroza, and I am a 14-year-old TKS innovator who has a really big passion for gene editing. And today we are going to be genetically engineering jellyfish bacteria to glow in the dark. So I hope you guys like it, and yep. So before anything, I want you guys to know that bacteria and all organisms need to make proteins to survive. And proteins are just tiny nanomachines that do everything from controlling our metabolism to keeping our heart beating. So they're pretty important. And in order to make a protein, a cell uses a DNA code. And for each 3 letters of DNA code is for a single amino acid. And proteins are just chains of amino acids. And this kit contains DNA with the gene that codes for the jellyfish GFP protein. This protein fluoresces and glows when you shine blue light on it. So essentially we will be inserting DNA into some bacteria, and this process is called bacterial transformation. So I'm taking my LB agar media and dumping its contents into a 250 milliliter glass bottle, and essentially this will make the plates in which my bacteria will start growing and form its colonies, and eventually will glow in the dark. But for right now, we are just making the plates, and this consists of using a 50 milliliter tube, which I will be adding 150 milliliters of water into the 250 milliliter glass tube, and I'm shaking it vigorously. And that was my cat dropping lots of equipment, and yeah, and I'm continuing to shake it until I have to microwave it for 30-second intervals until the mixture is fully dissolved. So you may be asking yourself, why should I care about gene editing? And I would say that's because it can literally solve the world's biggest problems. And I say this because we can gene edit crops and humans to be immune to cancer. And as for the crops, we can solve world's hunger crisis. So I think that's the reason why I'm so interested and invested into gene editing. But I mean, gene editing's really, really complex. And right now we are making our plates, so let's get back to the project. Right now I am going to start making my bacterial cell for transformation, and it does consist of non-pathogenic E. coli, so you don't have to worry if you get E. coli. For my bacteria, I added about 4 drops of sterile water from a disposable pipette into my E. coli bacteria, or my DH5A centrifuge tube, which is where the E. coli is, and then I had to shake it until I was sure that the bacteria was dissolved, and after that I do streak the bacteria that I just made into the LB agar plates that were sitting inside the fridge for about 2 hours. So 2 hours has passed and the plate should be ready, so let's see what are the results that we got. So now that both our bacterial transformation mix and our agar plates are ready, we can finally start mixing them together by using the little scalpel thingy that they gave me. And I tended to make little designs, but that didn't work out. So after I was convinced with the bacteria onto the plates, I then put them inside the fridge for about 2 days. So to see if my experiment was a success, I used blue light and glasses to see if my bacteria would go green. And as you can see in the arrows, you'll see the colonies will be glowing, and that means my experiment was successful. And I didn't film this, but when there was no blue lights being added, when the lights were just normal, you could see white bacteria growing on the plate, separately. And sadly I didn't a video of it, but we ended up making a successful experiment. Whoop whoop. Yeah, I hope you guys learned something here, and I hope I inspire some of y'all to look up gene editing. Bye.